Today I am going to tell you about cell cycle and DNA repair. What is cell cycle? It's the sequence of events taking place between two mitotic divisions to produce two daughter cells. There are four phases in each cell cycle: M phase, G1, S, and G2 phase. G1 phase is gap 1 phase, that is before the synthesis phase, and G2 is after the synthesis phase. Now this cell cycle, mitosis and these three phases comes under interphase. In mitosis, you have four phases: prophase, metaphase, anaphase, and telophase. These four constitutes mitosis, and interphase constitutes gap 1 phase, synthesis, and gap 2 phase. So all these together is a cell cycle. Some cells will enter G0 cycle, G0 phase. That is a state in which cells are viable and functional but are in a non-proliferative or quiescent phase. That is, they won't divide during this phase. For example, neuronal tissue, cardiomyocytes, etc. Now, this cell cycle should be regulated. Why the cell cycle regulation is essential? Because if it is not regulated, the mitosis will be going on dividing the cells. So bringing about multiple number of cells that will lead on to tumors or cancer cell development. So cell cycle regulation is very important. We can see how it is regulated. So this is the cell cycle. You have here mitosis, then G1, S, and G2 phase. You can see mitosis duration is very much minimal when compared to these three phases. So these three together forms the interphase. So the checkpoint for regulation is in between G1 and S phase, and here between G2 to M phase, and in mitosis at the metaphase there will be another checkpoint. Because when there is damage in DNA in the G1 phase, that is when there is errors in protein formation which help in replication in the synthesis phase. So the cell cycle will be arrested here, and after repair, this will go on proceeding to synthesis phase. So how this regulation occurs? The important proteins involved in regulation of the cell cycle are cyclin and cyclin-dependent kinases. These two together, they regulate the cell cycle. They tightly regulate the cell cycle. So cyclin, when it is combined with cyclin-dependent kinases, it gets activated. So cyclin-dependent kinases, they are at basal level throughout the cell cycle. But cyclins, they are a number of cyclins. Each cyclin will be increasing in concentration during each cell cycle. So when cyclin binds with cyclin-dependent kinases, they will activate some of the important proteins which are necessary for the progression to the next phase of the cycle. So they signal the onset of events of cell cycle. Now we can see here, these are the checkpoints. So here damaged DNA will be detected and repaired. Only when it is repaired, it progresses to the synthetic phase. So here incomplete replication errors are detected, and only when these errors are repaired, they will proceed to this. In G2 phase also, if DNA damage is detected, at this checkpoint it should be repaired, then only it will be progressing to mitosis. Mitosis is division of cells. That is, here only nuclear division and cytokinesis occurs. That is division of the cytoplasm. Here during this S phase, only replication of DNA. So here G1 phase, the cell grows. That is, all the proteins necessary for replication increase in number, increase in concentration in this G1 phase. So those proteins will be expressed in G1 phase. Then synthesis phase, during which replication occurs. Then in G2 phase, replication errors are detected and the accessory proteins which are necessary for the proper division of the cell cycle, mitosis, proper mitosis. Those accessory proteins will be formed here, increase in number, concentration here. So in G1 and G2 phase, all the accessory proteins which are essential for the next phase of the cycle will be increased in concentration, or will be synthesized, will be produced. So here cell grows, then DNA replicates to produce the dotted DNA, then accessory proteins for mitosis synthesized here. So here also checkpoint, then it proceeds to mitosis, and during metaphase, at the metaphase, if the improper spindle formation is detected, then cell cycle is arrested here. After repairing this, it proceeds to anaphase and telophase. Only then there will be proper division of the cell. So this is how cell cycle is regulated. So in each of the phases, there is proper combination of cyclin and cyclin dependent kinases. Specific cyclin and cyclin dependent kinases should be in each cycle. The cyclin and CDK combination at the beginning of the G1 phase will not be present in the S phase. So each phase has a specific cyclin and CDK combination. Only then the cell will proceed in proper phases of the cell cycle. So here you can see in G1 S phase interphase, cyclin D will be present. Cyclin D will be in increased concentration. So this starts rising during this phase. Then it binds with two CDK, cyclin dependent kinases 4 and 6. So this starts increasing. Cyclin D starts increasing from early G1 phase until end of the G1 phase. So when it reaches its peak, cyclin E starts increasing in concentration, because this cyclin E is essential for proceeding to synthesis phase. So this cyclin D and cyclin dependent kinases combination will increase the concentration, or will express the concentration of cyclin E. So they will activate the transcription factors necessary for cyclin E, and cyclin E will be formed, and this combines with CDK2 for the cell cycle to proceed to the synthesis phase. Next, cyclin E at the end of the G1 phase, cyclin E will be degrading and cyclin A will combine with CDK2, which helps in progression of the S phase. So during this phase, all the enzymes necessary for replication will be synthesized and increase in concentration, so that in the S phase, replication properly takes place. Then cyclin A increases during S phase and it begins to decrease at the end of the S phase, where cyclin A and cyclin B takes over for the cell cycle to proceed on to mitosis. Here you can see cyclin D combining with 4 and 6 cyclin dependent kinases. These are essential. This combination is essential for progression past the restriction call at G1 S boundary, or to proceed to S phase. And cyclin E and A combining with CDK2 is essential for initiation of DNA synthesis in early S phase. Initiation by cyclin E. Then as it goes on, as the level of E begins to decrease, A will bind to CDK2 so that it can terminate the S phase. Then cyclin B with cyclin dependent kinase 1 for transition from G2 to M. Finally, it will proceed to mitotic phase. So this is about cell cycle regulation. So now you know about cell cycle and how is it regulated, what is the importance of cell cycle. Now these are some of the regulators of cell cycle. So growth factors are essential for stimulating the cell cycle progression, like epidermal growth factor, PDGF, and fibroblast growth factor. And for inhibiting the cell cycle progression also, we need TGF, transforming growth factor beta. Then inhibitory proteins like CDK proteins, CYP and KAP proteins, and JNK4 proteins, all these are essential for inhibition of the cell cycle. Then proteases, these proteases are essential to degrade the cyclins. So these are some of the regulators. I'm not going into detail of each of these factors at this present slide. Next, DNA damage and repair of DNA. So environmental insults and replication errors will cause alteration in the bases or removal of some of the bases, which leads on to damage to DNA. So what are the various factors? Exogenous and endogenous. Exogenous caused by environmental insults like irradiation, UV light, etc. or chemical agents. And endogenous damage by metabolism and replication errors. So DNA damage may occur to either nuclear DNA or mitochondrial DNA. If this damage is unrepaired, that leads on to pathology, resulting in cancer, senescence, and apoptosis, depending upon the pathology. So what are the various types of damage to DNA? There may be single base alteration or two bases or chain breaks or cross-linkage formation. These are the various types of damage to DNA. So single base alteration may be found. These are the various causes of single base alteration, like depurination, that means purine bases have been removed. Then deamination of cytosine to uracil. So when cytosine is deaminated, when one amino group is removed from cytosine, it leads on to the formation of uracil. Likewise, deamination of adenine leads to the formation of hypoxanthine. Then alkylation of bases, insertion or deletion of the nucleotides, base analog incorporation, intercalators, like these leads to single base alteration. Then two bases alteration are produced by UV light or bifunctional alkylating agent cross-linkage formation. Chain breaks due to ionizing radiation, radioactive disintegration of backbone element, then oxidative free radical formation. Cross linkages formed between bases in the same or opposite strands, and between DNA and protein molecules like histones. So these are the various damages produced. Now, DNA repair mechanisms is very important because all the damages caused to DNA should be repaired appropriately, otherwise it will lead on to pathologies. So these are the various types of repair: excision repair, mismatch repair, direct repair, and double strand break repair. We can see one by one. So first one is direct repair, that is without excision. It repairs the damage caused by base modifications. So two mechanisms are under direct repair. One is by photoreactivation, another one is dealkylation of guanines. Photoreactivation is by the enzyme DNA photolyase. So this enzyme is activated by visible light. By photoreactivation, it cleaves the covalent bonds which forms the thymine-thymine dimers. So dimers covalent bonds are broken down by this enzyme that leads on to removal of the dimers and formation of the proper DNA. Then dealkylation of guanines. Here, guanine alkyl transferase enzymes are used here. They remove the methyl and ethyl groups from alkyl guanines. So alkyl group is removed by guanine alkyl transferases. This enzyme, it accepts the alkyl group which are removed from the bases and it undergoes ubiquitination. It gets degraded itself. So this is the uniqueness of guanine alkyl transferases. So it gets degraded, because enzymes usually they don't degrade, they don't change, they catalyze the reaction and comes to its original form. But here guanine alkyl transferase, it gets degraded by accepting the alkyl. So these are the two mechanisms of direct repair. Here you can see the photolyase activation. So UV light damage, this is the dimer formation. So covalent linkage is formed. So this photolyase enzyme through FADH chromophore, it cleaves the covalent linkage form and forms the proper base pairs. So it is repair. This is direct repair. Next one is mismatch repair. So mismatch repair, the mismatch base should be identified. So for this, the enzyme should know which is the newly synthesized strand. That is identified by the methylated bases. So for this, you should know this. This is the parental strand. So in parental strand, the adenine group will be methylated after replication. This is the parental strand. So methylation will be present, methylated adenine will be present only in the parental DNA, not in the newly synthesized DNA. So what happens here? Replication, this is the new daughter DNA strand formed. Here you can see the bases. This adenine is not methylated here. In the newly synthesized strand, adenine is not methylated. So this is how the enzyme recognizes the newly synthesized strand. So following replication for a short period, it will not be methylated, but after that it will be methylated by the methyltransferase enzymes. And after, once it is methylated, both these strands cannot be distinguished. It will be methylated. So after this, both the strands cannot be distinguished by the enzyme. So immediately after replication, this mismatch repair will be repaired, will be recognized and repaired. So you can see here. So this is the GATC sequences of which adenine is methylated. So one mismatch base pair is present here. The enzyme will correctly identify the mismatch base pair in the newly synthesized DNA. This is essential because which base is altered should be recognized. The base alteration may be in the parental strand or in the newly synthesized strand. So only newly synthesized strand has to be repaired. That will be recognized by the methylated adenine group present in the parental strand. So here the mismatch base pair is here. Mismatch means instead of adenine to thymine pair, adenine may be paired with cytosine. So this cytosine should be removed here. So here the reference molecule is methylated adenine in GATC sequence. So next, once the mismatch base is recognized by the enzyme, next thing, the single site strand cut by GATC endonuclease. So endonuclease cuts here, single site strand cut by GATC endonuclease. Next, the defect along with some of the nucleotides will be removed here. After the one exonuclease will come and remove all the bases. This is done by endonuclease. Then defect is removed by exonuclease. Exonuclease means it will act from the end, exonuclease. So defect is removed by exonuclease. And then after the removal of this, DNA polymerase will come and elongate the chains by complementary base pairing, as in replication. And finally, ligation by ligase enzyme. So first, endonuclease cuts the strand where near the mismatch base pair. Then exonuclease remove the defect along with some base pairs. Then DNA polymerase synthesizes the complementary base pairs in the daughter DNA. And finally, DNA ligase religate the phosphodiester bond. This is how the repair is done. Mismatched repair is done. Here, the main thing is mismatched base pair is recognized by the methylated adenine sequence in the GATC sequence. These are the various enzymes used in mismatch repair by the prokaryotes. So recognition is by mutase. Then this binds to unmethylated GATC, that is mismatch base in the daughter DNA, mutase. Then L binds these two proteins and H nicks the unmethylated GATC. After the nick, exonuclease will remove portion of DNA from the nick to beyond the error. From the nick, beyond the error. Exonuclease will remove portion, then DNA polymerase 1 fills the gap and ligase seals the nick. This is how it happens in prokaryotes. So in eukaryotes, same in a similar manner, but there are multiple enzymes present in eukaryotes. So here the mismatch repair, if it is not properly done, it will lead on to diseases, various diseases. One of this is hereditary non-polyposis colon cancer. This is most common inherited cancers. Here the mutation is human MSH2 and human MLH1 protein. So MSH2 is analog to mutase protein which recognizes the mismatched base pair. Then mutation leads to 50 to 60% of HNPCC cases are due to mutation of this MSH2. MLH1 is analog to MutL in prokaryotes. So how this faulty mismatch repair result in colon cancer? First, detection of the microsatellite instability, that is, if it is not repaired, it will lead on to the microsatellite formation, that is, repeated small segments of base pairs. So microsatellite will be formed. The mutated proteins of the mismatch repair enzyme is present in these individuals. If it is present, it will not repair the microsatellite bases. So unable to remove the small loops of unrepaired DNA, unpaired DNA. So increase in the microsatellite DNA expansion. So this affects either the expression or the function of a protein, which is critical in the cell cycle regulation of these colon cells. This is how it leads to non-polyposis colon cancer. Now next one is base excision repair mechanism. Here the enzymes involved are specific N-glycosylases, which recognizes the depurinated site. Here base excision. So one of the base is altered and that is removed. So recognizes the depurinated site and then removes the base. So this marks the site of the defect. Then this allows, because of the removal of the base, there will be production of apurinic or aprimidinic site depending upon the base which is removed. Then corresponding specific endonuclease acts on this to excise the A basic sugar. Then the proper base is replaced by DNA polymerase 1 as in the previous case, and finally ligase returns the DNA to its original state. Here you can see base excision repair. So this is the DNA. So because of heat energy, spontaneous deamination of cytosine to uracil occurs. So here in this DNA, because of the heat energy, the cytosine undergoes spontaneous deamination to uracil. So cytosine forms uracil, but uracil is not normally present in DNA. So there will be instability here. So similar deamination will take place for adenine and guanine. Adenine lead on to hypoxanthine and guanine forms xanthine. So once uracil is formed, the specific DNA glycosylase recognizes it. So uracil DNA, here uracil is formed. So uracil DNA glycosylase recognizes the formation of uracil. So this glycosylase, this hydrolyzes this bond, removes the uracil. So the base is removed here. So what happens? Base is removed. This produces A-purinic or A-pyrimidinic site, AP site. So here the uracil, so A-pyrimidinic site is produced. Next what happens? Endonuclease, the corresponding specific endonuclease acts on this. It removes the bases. So 3 bases are removed here. Base excision repair. So base is removed. Next, so this is synthesized by DNA polymerase, and DNA ligase synthesizes the complementary base first in the damaged DNA strand. So this is how base excision repair occurs. So first deamination, because of deamination, which is produced by heat energy, one of the base is deaminated, producing uracil. That is faulty base pair is the base is produced. Then this faulty base is hydrolyzed by the specific DNA glycosylase. AP site is produced. So this sugar is removed. So AP endonuclease removes the sugar along with the phosphate bond. So here AP site is produced. Next what happens? DNA polymerase and ligase synthesizes the broken strand as in the previous case. This is how base excision repair occurs. Next one is nucleotide excision repair. Here replace regions of damaged DNA up to 30 bases in length. So it is nucleotide excision repair. So this is caused by UV light or smoking or ionizing radiation, cancer therapy, chemotherapeutic agents, and a variety of chemicals. This leads on to base modification, strand breaks, cross linkage between bases and between DNA and protein, and numerous other defects will be produced by these. So here we can see how this nucleotide excision repair occurs. So here, because of the UV light, there will be dimer formation here. So human Xc nuclease acts on this. This is in eukaryotes. Human Xc nuclease acts on this. So this removes some of the bases from the 3' end and some bases from the 5' end. So longer length in the 5' side it removes. So here this is the damage caused. So this nucleotide, around 29 base pairs are removed here. Nucleotides are removed. Oligonucleotides is removed by human Xc nuclease. So it removes the nucleotides on either side of the damage. So that is removed. Unwound by DNA helicase. Xc nuclease puts the nick on either side. So human Xc nuclease produces nick on either side of the damage produced. Then DNA helicase removes that part. After that, as before, DNA polymerase and DNA ligase synthesizes and ligated the DNA strand. So this is how nucleotide excision repair occurs. Here the enzymes are human Xc nuclease produces the nick here. And then DNA helicase removes this part. And then this part, removed part, is repaired by DNA polymerase and ligase. The disease clinical significance of this is this faulty nucleotide excision repair leads on to xeroderma pigmentosa. The first disease to be caused by defective repair of DNA. Defect in the repair of damage to DNA caused by UV rays. So absence of the enzyme UV specific endonuclease will lead on to xeroderma pigmentosa. The clinical features are sensitivity to UV rays, hyperpigmentation, blisters on the skin, leads on to cancer. So other disorders here are ataxia telangiectasia, Fanconi's anemia, and Cockayne syndrome. So this is xeroderma pigmentosa. You can see multiple pigmented and atrophic areas of face with occasional solar keratosis. And this is advanced stage of the disease. You can see the damage produced here, destruction of the eye here. So next one is double strand break repair of DNA. Here two systems are available here. One is non-homologous end joining repair and the other one is homologous recombination repair. Non-homologous end joining, what happens here is the ends of the two DNA fragments, they are brought together by a group of proteins which help in their religation. So this non-homologous end joining is simplest but least accurate. So this is error prone and is mutagenic. This method of repair is error prone and mutagenic. The proteins involved in this is Ku protein, which is the ATP dependent kinase, and DNA dependent protein kinase. Defects in this repair will lead to cancer and immunodeficiency syndromes. I'm not going in detail about non-homologous end joining in this slide. So here, this is the simplest overview of non-homologous end joining. Here you can see the double strand break here. So Ku and DNA PK proteins, these two proteins bind to both the ends, both the broken ends. So they brought these ends together. So you can see here, approximates the ends. Then lead on to the unwinding of one of the strands, unwinding here, and then form the complementary base pairs here, and the excess will be removed and these will be approximated. So this is how ligation of this occurs. This is how non-homologous end joining, because they are non-homologous in the same chromosome, double strand break occurs. So for synthesis, if the double strand break occurs, for synthesis, they need complementary strand, template strand. So here, these two proteins approximate these ends and unwind the ends so that one of the strand will act as a complement for base pairing and then this joins. Finally, joining of the phosphodiester bond is by ligation. So this is how non-homologous end joining occurs. Homologous recombination is similar to recombination that occurs during meiosis. So this uses the enzymes which normally perform genetic recombination between homologous chromosomes during meiosis. This is most accurate, much less error prone. That non-homologous end joining is least accurate, that is error prone and mutagenic. Here there will be exact reconstitution of original sequence using homologous chromosome as a template. So this is dominant method. This occurs in S and G2 phase after sister chromatid is created. The proteins involved here are, some of the proteins are RAD52 and RAD51. So here so many proteins are involved in eukaryotic recombination like mRNA complex and ATM proteins. So much of proteins are there. I'm not going into detail. So this is overview of homologous recombination. This is how this occurs. So double strand break occurs here. This is the homologous chromosome. These two are homologous chromosomes. So here, this chromosome, which is intact, will act as a template for the synthesis of this strand here. So that is homologous recombination. Here what will happen, when there is double strand break, these proteins which are essential in recombination will bind and they will reset the ends so that 3' overhang is present. So here you can see when this is When this end is cut more than this, there will be 3' overhang. Similarly here, 3' overhang is present because this is resected up to this part. So here this is 3' overhang. Next what will happen, this protein causes this overhang to invaginate into the homologous chromosome. So this end invaginates here into this double strand. So because of this invagination, this will goes like this. So here this will scan all through this template. So this acts as a template. So this invaginates here and this protein scan this strand till the complementary base pair occurs. So next, after the complementary base, so it directs the strand deviation and this is the D-loop formation. This is termed as D-loop. Next, strand extrusion. Because of this invagination, this strand becomes extruded. Next one is second end capture. That is, so here this strand is synthesized using this strand as the template. So here this is newly synthesized. Similarly, here this will be extended using this strand as the template strand. So both of these will be extended and then they will be recombined. There will be a cut here. They will be divided. So this is how homologous recombination occurs. Next, other diseases caused by defective repair are ataxia telangiectasia and Fanconi's anemia. So this is an autosomal recessive disease leading on to cerebellar ataxia, lymphoreticular neoplasms. Fanconi's anemia is also autosomal recessive anemia. There will be increased frequency of cancer and chromosomal instability. This is due to defective repair of cross-linking damage. So finally, how this repair occurs, damaging agents are X-rays, oxygen radicals, alkyl agents, lead on to uracil, a basic site or 8-oxoguanine or single strand breaks. These are the lesions that will be formed by these agents. So this, to rectify this base excision repair. Similarly, UV light and polycyclic hydrocarbons lead on to photoproduct formation, bulky adducts, cyclobutene pyrimidine dimers, and the repair here is nucleotide excision repair. Similarly, infrared, UV light, X-rays and some anti-tumor agents leads to interstrand cross-link damage or double-strand breaks, which will be repaired by recombinational repair, either homologous recombination or non-homologous end joining. Then replication errors, mismatch errors, aging mismatch or TC mismatch, insertion or deletion, all these will be repaired by mismatch repair. This is how repair is done. This is one of the example for ataxia telangiectasia. This is in conjunctiva and this is in ears. You can see the new cell formation here. So with this, I am finishing off cell cycle and DNA repair systems.